Good morning, everyone, and welcome to the BioVexo cluster webinar, Innovative Microbial Solutions for Plant Health, Development, Field Application, and Regulatory Integration. I am Ben Leitner. I'm the Dissemination and Communication Manager for the BioVexo project. This webinar will be recorded, and you will be able to access it on the BioVexo website shortly. Today, we will hear insights on sustainable microbial solutions for plant health and how this innovative approach can find its way into the market. It's my pleasure to welcome the sister projects BeXyl, represented by Ms. Blanca Landa, Project Coordinator, NextGenBioPest, represented by Ms. Angeliki Miliotti, and Life Resilience, represented by Mr. Vasco Abreu, Commercial Director at NutriPrado. From BioVexo, I'm happy to introduce Mr. Stefane Compant, Scientific Coordinator for the BioVexo project, and Mr. Pasquale Saldarelli, Work Package Leader for Field Trials, also in the BioVexo project. After a brief introduction to the respective projects, we look forward to a panel discussion, followed by an open discussion where I encourage you to share your thoughts with our experts. And please write your comments or questions in the chat, and we'll try to accommodate them all. In today's webinar, we hope to shed light on the enormous potential of microbial solutions to strengthen plant health in the light of plant pathogens, on successful practices in developing microbial solutions, highlighting characteristics on field applications of microbial solutions, and explore challenges for microbial solutions in the EU regulation process. I would now like to hand over to Mrs. Francine Martin, Project Manager of the EU-funded BioVexo project. Thank you.
Thank you, Ben. Thank you. Hi, everyone. Good morning and welcome to this webinar. My name is Francine Martin. I am the BioVexo Project Manager working at RTDS Group. I'm really happy to kick off today's meeting, cluster webinar. And first of all, big thank you to everyone who's here today, who take your time to be here today, especially to our speakers from BeXyl, Mrs. Blanca, NextGenBioPest, Ms. Angeliki, and Life Resilience with Mr. Vasco Abreu. And it's great to have you here with us today. I really appreciate for taking your time to be here. And also a quick shout out to my colleague Ben Leitner, also from RTDS, who did a fantastic job to organize this webinar today. Thank you, Ben. And just a few words, I would like to introduce you a bit very quickly to BioVexo, so you can just get a bit of a context. BioVexo is a European project. It's funded by the Horizon 2020 under the Bio-Based Industry Joint Undertaking. We are a team of 11 partners from 5 different countries, all working together to develop these biocontrol solutions. And just a bit of these 11 partners, we at RTDS and also the Austrian Institute of Technology, we're based in Austria. In Italy, we have CNR with Pasquale, who was just introduced yet now, and also CRFSA. And in Spain, we have the University of Sevilla, DOMCA. In Belgium, we have the University of Antwerp and GlobalCam. And in Slovenia, we have ACES Bio. So yeah, we are all these 11 project partners. We started in May 2020. We were originally planned to finalize the project in April 25, but we finally we're very excited that we got this 8 months extension to continue until December this year. And this also gives a bit of a time for us to explore and fine-tune our results and solutions. So today's webinar is also part of this joint extended journey that we wanted to take and to bring effort to bring all these projects together. And we really hope to give this space a chance to share all the results. And we're also thrilled to invite you all for our final BioVexo event, which will take place in person. It will be in November 13, which is a Thursday. It will be in Apulia, Italy. It's a full-day event. packed with presentations, panels, everything. Also, this chance to connect face to face and finally meet one another. And yeah, we will post everything soon in our website regarding registration, agenda, and logistics details. And follow us on our social media and stay tuned for news. So now I would like also to give the floor to our scientific coordinator, Dr. Stefan Compan. Stefan is a leading scientist in AIT. He's a specialist in plant microbiome, and he's guiding us in BioVexo since the beginning of the project, and will walk us through a bit on the project's journey so far. So thank you again for being here today, and I hope you enjoy this great session.
Thank you very much, Francine. I just need a few seconds to share my screen and to find my presentation. So good morning, everyone. Just let me 1 second. No. Yeah, I think you can see now my screen. Just a moment. It's a bit slow in the morning Zoom.
Yes, we can see the screen. Thank you,
can see the
Stefan.
screen. Yeah, so good morning, everyone. I will talk about BioVexo. There was a very nice introduction from Ben and Francine from RTDS. What we are doing in BioVexo is to develop some solution to control Xylella fastidiosa on olives and almonds and the insect vector for integrated pest management. As Francine said about BioVexo, it has started in May 2020, should have ended in end of April this year, but we asked for extension. So it's a 5 years and half a years project. It's granted by, funded by Horizon 2020 with a bio-based industry consortium. And as Francine said, there is 11 partner with industry, SMEs, academia, farmer association from 5 different countries. What we are doing in BioVexo is to try to develop some solution to control the disease you have in front of you on this slide, caused by a phytopathogenic and quarantine bacterium, Xylella fastidiosa, which is now present in Portugal, Spain, France, Italy, Corsica, and also in Israel. You have different kind of species and different strain in Europe. And this strain is transmitted by an insect vector. What you can see here is this Philaneus plumarius, 2-3 millimeter long, which transmits this bacterium to the plants and cause a disaster, as you can see on these photos. Up to 100,000 jobs has been already lost. This cost a billion of euro, and there is no solution at all on the market. So it's a catastrophe for cultural heritage, for farmers, for the economy, for the social behavior, and it's really making a collapse of olive trees in Puglia, but also almonds in Balearic Island and also in Alicante. What we do in BioVexo is, I told you, is to try to develop some solution against the insect vector or against the phytopathogenic bacterium itself as preventive or curative use. At the beginning of the project, we chose 6 biocontrol solutions, but in fact, it was 8 solutions. There were some bacterial strain targeting Xylella fastidiosa, microbial metabolite against the vector, plant extract against Xylella or against the vector, and an anthropogenic fungi. We did not start from scratches at the beginning. We chose this solution because they were already developed in our pipeline from different company or different academic partner. And we wanted to develop this solution to be close to the market. So you imagine it was a lot of work, and it's still a lot of work to do before end of the project. But first, we made tests in small-scale field trials to see if some solution works or not. Against Xylella, against the insect vector, and in different places in Puglia, in Italy, but also in Majorca and a bit in Alicante. Then the next challenge was to formulate the biopesticide and to upscale their production before to do large field scale trials and IPM strategy. You can imagine that we treat a lot of plants in the field and we needed an upscale solution and formulated biopesticide to store them and to apply sufficient amount in the field. And in BIOVEXO, we also studied the mode of action of some biopesticide. I mean, if some biopesticide give positive result, we want to understand how they work. What are the effects? How are they, it's possible that they reduce, for example, the symptom spread. And in BIOVEXO, we also analyze the sustainability of our product, the toxicity and toxicology analysis, the non-target effect on bees. So we wanted to develop, and we still want to develop, some sustainable solution that can help farmers, society, to reduce the Xylella pressure in the field. Of course, in an industrial point of view, we need that the biopesticides has a good life cycle assessment, meaning that the production should not increase the production of carbon dioxide, etc. It should be sustainable. And what we obtained so far, we had upscaled and formulated some biopesticide. We performed tested on olive orchard and almonds against Xylella and its vector. We tested different mode of application according to the biopesticide, foliar spray, drip irrigation, but also endotherapy. Life cycle assessment was done, the mode of action was partly determined, and the toxicology and toxicity tests were done. And we have passed from component research and development at year 4-5 at the beginning to end of the field trial and to be close to the market. Now, the fields. Pasquale Saldaridi, here present, together with Enza Don Giovanni from CRSFA, made the planning to make large-scale field trial in Puglia, in Majorca, and in Alicante. In Alicante, there was a problem. It's an eradication zone, so we could not continue our test in the field. But we have done tests in Puglia and in Majorca, and test after application of different biopesticide every 6 months, the population of Xylella fastidiosa by quantitative PCR in preventive and curative approach, but also if there was a reduction of the spread of the symptom. What we got so far is that no biopesticide at all reduced the Xylella population in plants. There was some trains, but it was not significant. So no solution reduced the Xylella. But if you see here the area on the disease progress curves, untreated plants from 2023 to 2024, and plant treated with biopesticide 1, there was a slowdown of the progress of the symptom in the field, in curative trials, in San Vito de Normani, when we analyze block of 56 plants per treatment after 8 applications per year. The biopesticide 2, there was no effect at all, but with the biopesticide 1, there was a reduction of the progress of symptom of 46.5%. You can tell me, what is this biopesticide? In fact, it's a gamma protobacterium. It's a Parabiocordia phytopheromone strain PAGN. We applied this strain after application of the formulated product done by SS Bio in the field as foliar spray. And the biopesticide 2 was onion extract developed by the company Domka. It was applied as a drip irrigation. Why we choose this formulated PAGN? It's because a few years ago, Stephen Lindo from California shows the potential of Paragorgia phytopheromone strain PAGN to protect grapevine disease against Xylella, and there was positive result in the field. So we choose this strain to test it on olives and almonds, and there was a nice result. If we check across year in San Vito de Romani, where we have done the test, where the plants were 3 years old, we follow the severity of symptom in untreated plants, formulated PAGN treated plants, onion extract, on new symptom and old symptom. And the disease curve from March 2023 to March 2025 on new symptoms, there was a reduction after application of formulated PAGN compared to untreated plants, and also on old symptom. Across year, compared to the control, there was 53.7% reduction of the spread of the symptom on all symptom of olive trees to up to 28.1 in March 2025 with formulated PAGN. With onion extracts, it was not significant. On new symptoms, there was also a reduction with formulated PAGN, passing from 44.9% of reduction of the spread to 31.9% of the spread. Problem when you go to the field, it depends on the location and the year of the plants. We have done another test in Puglia, in Oria, with plants aged of 30 to 40 years old, and we follow the severity and the progress of new and old symptoms. The problem, there was in October 2024, no reduction of the spread of the symptom when we analyze the plants treated with formulated PAGN compared to untreated plants on new symptom and old symptom. There was a strong severity, in fact. You see here the McKinney index for the severity. We are quasi at 70% on new symptom, but also old symptom. What we have seen in this field at the beginning of the experiment, there was a positive reduction of the spread of the symptom, 20% on all symptom in Oria to 15%, and then it was not significant because of this too strong severity. The same with new symptom. We know that with biopesticide, where the severity is too strong, there was no positive effect. It might be the case here. We still continue the analysis of these field trials. I did not show you also in Mallorca some results. For the moment in Mallorca, we don't have positive result, but you know, when you apply biopesticide, it takes years before you get a reduction or not of symptom. What we have seen also in BIOVEXO, after doing integrated pest management strategy, where we planted some olive plants as preventive and combined the application of biopesticide targeting Xylella or the insect vector and comparing this to chemical product or the product, what we have seen so far. There was not yet no symptom on Xylella on this plant, but what we have seen in Francavilla, that there is a biopesticide targeting the insect vector can reduce 50.19% of the adult vector compared to the control. Also on the juvenile insect, 54.2%. It was in Francavilla. In Squinzano, there was a problem. There was a too low population of spittle bugs. We use stick traps and sweep net for that. It's just the result we have now, but we still continue until the end of the project. I told you before, when there is some biopesticide having positive effect or negative effect or neutral effect, we want to understand how they work. I will not pass too much time for this slide. What we study is the effect of the biopesticide, for example, formulated PAGN, on the plant transcriptome. What we have seen so far, there is an induction of the plant immunity and also induction of gene related to plant growth promotion. And this is what we have seen also in the field. We saw reduction of symptoms, for example, in San Vito de Normani, but we have seen also an increase of plant vigor of the plants in Mallorca, but also in Italy. When you want to develop a biopesticide, it depends on the claim when you want to put this in the market. What we have seen with some biopesticide, we have biostimulant effect and also biopesticide effect. We are studying also the effect of the different biopesticides on the plant microbiome. Why? It's because some biopesticide might modulate the microbiome of the plants and to indirectly an effect against Xylella. We are studying also the effect of biopesticide on the insect microbiome and the evaluation of persistence of formulated PAGN, for example, in the environment. In the environment, we have seen that formulated PAGN after 8 applications per year and 1 to 2 months after the last application, there is only a minimal presence of the strain on olive plants. It doesn't survive well. But this doesn't matter because if it provides positive effect and doesn't persist in the environment, this is very good for sustainability. So in a nutshell, to be in a summary, what I show you today, sorry, I did not show you too many slides. There was only 10 minutes for that. We formulated PAGN, Parabiocolera phytopheromone strain PAGN. What we have seen so far, it slowed down symptoms on young plants. We know also that improves tomatal conductance. I did not show you slide for that. We know that it's not an under fight on olives after foliar application. It improved plant vigor, induced plant immunity, and plant growth-related genes. We have seen the effect in Puglia, not yet in Mallorca. And for onion extract, we have seen only biostimulant effect. We have not seen a reduction of symptoms, but we have seen an improvement of plant vigor. And for the biopesticide targeting the insect vector, what we have seen so far and in the field, reduction of the juvenile and adult population. You will see this project is 5.5 years project. It's very long project. But when you want to be close to the market, there is a lot of things to do, from the lab to the field that come back to the lab. And there is a lot of things to do and also to make field applications. There was a lot of field tests and on a lot of plants in different locations. If you are interested about the project, don't hesitate to ask us for questions or also to visit our website, www.biovexo.eu. Thank you very much.
Thank you, Mr. Compant. This was really interesting. Thank you. I would now like to ask Mrs. Lander to share her presentation and tell us more about the BeXyl project.
Let's check. Can you see now? Can you see my presentation?
No, we cannot yet see the presentation. Yes, now we can see it. Thank you.
Okay, thank you so much. I will try to go fast within the 10 minutes. So I will present BeXyl project, also the main activities we are conducting related to bioproduct development. So as you know, Xylella fastidiosa is the pest most regulated within the European Union. It's the first priority pest. And since different outbreaks were detected in the European Union, many projects, European projects within Horizon 2020, were developed to try to fight against this bacterium. In 2021, after finishing some of those main projects, we decided that was time to continue working and doing research on this bacterium. And I have to say to you that the outputs of most of this project has contributed a lot to develop the different decisions and regulations within the European Union. So it was a very good example of how science contributes to develop policies. And currently, there is a request within the European Commission to EFSA to update the scientific opinion on Xylella fastidiosa, in which BeXyl project is contributing a lot. So in 2021, we saw this call in Horizon 20, in which we should target a quarantine pathogen. So we decided that Xylella fastidiosa was the best candidate to focus only on it. These were the objectives that the call should focus on. Another important aspect was we should envision international cooperation within affected countries, and also that the project should be a multi-actor approach. So for that, in the EFSA workshop in 2021, and after receiving all the outputs from the different projects, we considered that there were still many research needs from farmers, nurserymen, risk managers, and legislators. So we designed this project, BeXyl project, to answer these research needs. And we developed different scientific objectives. I'm not going to read them. You have this information in our webpage. So the idea was try to progress and focus on and implement the research output from this project and moving on and advancing in the knowledge of fighting against this pathogen. So our consortium is formed by 31 partners. There are 28 beneficiaries from 14 countries, from Europe, and also from other countries where Xylella fastidiosa is present. And also we have 3 associated partners, such as UK and Australia. It was very good to put into use the expertise of partners from the American continent when the bacterium is present since the beginning, and also because they can perform the experimental approaches more easily as we can do in Europe, in which we need to use biosecurity facilities or just work in the field, but only in areas under containment. As Stefan mentioned, it's not possible to do experiments under field conditions in areas under eradication. This is a 4-year project, and it will finish at the end of August next year. Another important aspect is a multi-actor approach. So we have more than 45 entities enrolled in the project, in which we try to absorb all the diffusion and communication aspect of our project, and also they participate in some activities that are essential for the correct project development. So this is the main scheme of our project. We try to answer 3 main outcomes and give impacts to the European Union in this aspect, to enhance capacity to prevent, monitor, and find adequate responses to Xylella fastidiosa, to enhance the capacity to control the bacterium, but mainly by biological approaches, and then to provide scientific-based support for relevant European and associated countries' plant health policies. And the main difference of this project is that for other projects, previous projects, the main focus was olive crop, but now after the different outbreaks appear in Spain and in other areas, so we saw that also Xylella was a big problem for almonds and for grapevines in Europe, and also for the natural environment. So concerning the first impact, we are trying to understand the drivers of Xylella fastidiosa epidemics, including climate change, We focus a lot in developing new improved methods and strategies for early detection and disease monitoring, not only molecular approaches, but also new trap insect, different modeling, remote sensing, and also even sniffing dogs for early detection. We are also testing the effect of sanitation using thermal treatments for propagating material at nursery and quarantine level. The second impact that is the most important for this workshop is to enhance capacities to control Xylella fastidiosa. So we are trying to implement all the different management approaches for implementing integrated pest management systems, focusing not only in biological control, but also in testing new chemical molecules. And one aspect of this approach is that we are targeting the three components of the pathosystem. So we are devoting one work package to develop genomic tools linked to finding resistant cultivars. We are also testing the vector by trying to find environmentally solutions for effective control, or for not only for killing the vector, but also to disrupt the feeding behavior of the vector. That is quite important. And finally, of course, we are focusing also on the bacterium in the plant, trying to target different approaches, as you will see later. So concerning the host plant, that I also consider that is also a biocontrol measure for the bacterium. So we are continuing with the previous experimental plots developed in other projects, because Xylella infection takes many years for the plant to develop symptoms. So this is a long process that needs time and continue monitoring over the years. Also dipping into molecular responses to Xylella fastidiosa infection. Mapping genes associated potentially with the resistance that the lechino cultivar shows to the plant. And also we are trying to develop new progenies of olive crop, developing new crosses of resistance with other crops of interest of the varieties. Concerning the biocontrol of the vector, we are exploring entomopathogenic fungi, viral parasitoids against the vector, but also evaluating the efficacy of ground cover management, trying to use trapping plants or plants that are not as good for the vector to feed. Concerning entomopathogenic fungi, you see here different strains that were very effective in vitro for killing the vector. And the approach is being followed mainly by the University of Cordoba and also by the University Aldo Moro. They are trying to target the population of the vector at the nymphal stage when they are in the grass. Also when it start moving to the trees, when it develops wings and can fly, and also when it's feeding on the canopy of the different trees. So the approach is to develop different strategies. You can have the application of the entomopathogenic fungi directly into the vector, but also on the grass, so it can have a direct and indirect effect of both the nymphs and also the adults. But also there is an indirect effect of the vector, because some of these entomopathogenic fungi are endophytes. So it has been shown that when the vector feeds on the plants that are already colonized by these entomopathogenic fungi, the feeding behavior changes dramatically. So this can interfere with the Xylella fastidiosa transmission. That is an additional effect on the vector. So concerning the effect and the use of biocontrol tools against the bacterium, we are developing synthetic microbial communities of Xylella inhabiting bacteria. So we are doing a selection of both in silico. We have the genome of this bacteria. We are doing many tests in vitro and also in planta. So we are selecting from a collection starting for over 300 strains. We develop different tests to select those bacteria that has more positive direct and indirect biocontrol mechanisms, such as production of antimicrobial compounds, chitinase, different enzymes, etc., that were able to also inhibit the pathogen in vitro, and also that they were compatible among themselves. That is quite important when you want to develop a community of different bacteria. Now we are testing in the plant, not only in the greenhouse, but also under field conditions. And so far we have seen a reduction of the inoculum of the bacterium of Xylella present in the xylem. We are testing also new molecules, antimicrobial compounds, mainly plant extracts derived from the olive oil industry, but also we are testing antimicrobial peptides that has a different effect on the plant, not only target Xylella and have antimicrobial and antibiophil properties, but also the elicit plant defense response in the plant. And they have been shown under greenhouse condition very effective results that we are now testing under field conditions in Mallorca on almonds. Another approach is the use of bacteriophage that are phages that are able to kill directly Xylella fastidiosa cells. Different experiments in planta showed a drastic reduction in Xylella fastidiosa titers in the inoculated plants. And these bacteriophages are being tested now in Mallorca on grapevines, and so far with very interesting results. And just to finish, one of the main aspects of this project, because it's a multi-actor approach, is to provide scientific-based support for relevant European associated countries. As I told you, we have developed different socioeconomic and ecological impact analysis, and we are providing some of the results that are not still published in scientific papers are being transferred to EFSA for the updating the scientific opinion. And if you are more interested, you can visit the website in which you have the different practice abstracts. We have also a newsletter that is monthly published. We have different documents that are open access. Also, all the scientific papers are published in our webpage. We have very active in social networks, and we develop, starting again in September, every 2 weeks, scientific seminars related to Xylella fastidiosa work. And that's all from my side. And I want to thank all the consortium, BeXyl. What I tell you today is the work and the effort of many, many different partners. We are over 100 people participating in the project. Many young scientists that are taking knowledge on this bacterium, and I hope they will be the relevant person in the future to continue fighting against this bacterium. Thank you.
Thank you very much, Mrs. London. This was very interesting, and I look forward to hear more from you during the panel discussion. I would now like to ask Ms. Migliotti to present NextGenBioPest. For this purpose, I will share my screen. 1 second, please. Okay. Okay. So, Ms. Migliotti, you have the floor, and please just tell me when to go to the next slide.
Yes, thank you very much. So let me introduce myself. I'm Angeliki Migliotti. I'm Dissemination, Exploitation and Communication Manager at Smart AgroHub and at NextGenBioPest. Thank you for today's invitation. NextGenBioPest is a sister project with BIOVEXO. So we have executed several clustering activities together. First, I will tell you some things about Smart AgroHub very briefly, and then I will present you our project, NextGenBioPest. I will try to be very brief because I think we are running out of time. So next slide, please. Okay. So Smart Agro Hub is actually a spin-off of the Agricultural University of Athens. As a company, it was established in 2021, and we are comprised of 10 shareholders. These shareholders are companies of the agri-food industry, and also one of them is the Agricultural University of Athens. Currently, we are more than 20 employees, in-house and external collaborators, and now we are in the peak of our projects. We are involved in more than 25 European projects currently. We are active in Horizon Europe projects, Digital Europe projects, and Erasmus+. In Horizon Europe and Digital Europe, we are leading the dissemination communication work packages and the exploitation work packages, while in the Erasmus Plus projects, we have also coordination roles in some of them. And generally, in some of the projects, we have some technical involvement, but not in all of them, because we have other services as well in Smart Agro Hub. For example, we have some smart farming tools, and we are involved in agricultural experimentation in a field in Greece called Liartos. So there we have some fields, and we execute several experiments about smart farming. We use sensors, drones, etc. Also, we offer some sustainability consulting services like life cycle assessment. This means that we collaborate with food industries, for example, in order to define the carbon footprint their products have, because this life cycle assessment is generally asked nowadays, and industries are very interested to know what their what are their emissions. And also we have the Smart Agro Lab, which is a new initiative. This is actually an incubator. We are in contact with several people that have some ideas, innovative ideas, and we help them to make these ideas actually companies. Or in case they have already a company, a new company, a startup, we help them through mentorship to make it evolve. Next slide, please. So let's pass now to NextGenBioPest. On the next slide, I think I had put the teaser video, but let's skip it for now because we are running out of time. So next slide. Okay. NextGenBioPest is funded under the Horizon Europe program. It is under the cluster 6, and it is an innovation action, which means that we are actually going to develop some products and to do some research. The EU contribution was around 6 million euros, and the project started in March last year, and it will end in February 2028. We were 22 partners from 11 countries, but we made an amendment last month, and now we are 24 partners. One university was added and one startup as well. The coordinator is Fourth Foundation of Research and Technology. It is based in Heraklion, Crete, and the person in charge is Professor Vontas, John Vontas. And as I said, in Smart Agro Hub, we lead the dissemination, exploitation, and communication work package. The partners are from Greece, Serbia, Canada. We have partners in Germany as well. Generally, we were trying to build a consortium, a multi-actor consortium. So for this reason, we have universities, companies, institutions, and several organizations. Next slide, please. Here you can see very briefly the specific objectives of our project. First of all, we have developed a forum, an online forum, for which I will talk you later on, in order to capture the needs of the actual market and of the actual stakeholders. Also, in terms of product development, we are going to develop, and we have already started to develop an application which will give the opportunity to the farmer to take some photos of the plant and know what pest has damaged the plant before the symptoms are very obvious with the naked eye. Also, we are going to use some biological control agents. The main product of the project will be some RNA-based pesticides. Also, we are going to promote generally to the farmers the concept of integrated pest management. And we are going also to work on the social, economic, and environmental aspects of these new biopesticides. For example, we are going to, we have some deliverables regarding the regulatory background about these products, etc. Next slide, please. So, as I said previously, we have the stakeholders forum, but let me explain to you better. As NextGenBioPest, as all projects, we have a website. In the website, there is an innovation hub. The innovation hub is actually a place where additional information is added about IPM, RNA pesticides, sister projects, etc. In this forum, in this hub, you can join in two ways. If you are a person, I mean not an organization, if you are just a person, a student, a scientist, a farmer, you can register with your email or through Google and Facebook account. And then you unlock the additional content, the educational content that is produced in the project. In case you are an organization, a stakeholder, you can join by sending us your logo. We add the logo to the forum. Until now, we have 51 stakeholders and 120 registered members. So these stakeholders, as a first step, of course, they are showcased because we have created this network. We have collected several logos. So everyone who joined there can see other similar stakeholders in the field. And then, as more practical steps, these stakeholders are actually participating in the project through our events. They are invited as speakers, for example, many times, or they are asked to contribute to a deliverable, or they give some best practices, etc., etc. Next slide, please. And here you can see an overview of the content that is added in the hub. You see the login button, it is for the members, I said you previously. And as you can see, you can have access to several upcoming workshops, not only from NextGenBioPest, but generally workshops relevant to the field, some job openings, some training courses, and some funding opportunities. Next slide, please. And here you can see the stakeholders forum, just a small picture of it, how the companies are showcased. And also you can see the knowledge repository, which is the place where we collect all the presentations, for example, from the events that we have organized as educational material for other projects. Also, we are very active in social media. As you can see, we are present in many social media. So I would like to kindly ask you to follow us and stay tuned for our progress. Thank you very much.
Thank you, Ms. Miliotti. That was very interesting. I would now like to ask Mr. Abreu from the project Life Resilience to enlighten us on what Life Resilience is actually engaged with. So you have the floor.
Now is Ben. Is good in your screen?
Yes, looks very good. We have full screen.
Okay. The Life Resilience Project is a project of 3 countries, Italy, Spain, and Portugal. And thanks to Ben, thanks to another webinars, Francine, Pascal, Stefan, Blanca, is very interesting is compartilhar this. I think that a phrase of my friend Albert Einstein says, insanity is continuing to do the same thing over and over again and expecting different results. And now I expect my presentation. What is Xylella fastidiosa? With now in this webinar is a pathogen bacterium cause disease in different words. Is detected in 2030 and obviously in short authority in Puglia, no? Is many, we speak many things of Xylella. I go to objective of this project. Prevent and mitigate the spread of through a more resilience and sustainable quality production system. A performance with between olive varieties, evaluate offspring and select potential new varieties resistant to Xylella fastidiosa. These new genotypes will constitute conservation alternatives for the areas and extreme value resource in the case hypothetical advance of this disease. Demonstrate that sustainable farming practices include natural vector methods will help prevent the spread, make intensive plantation less vulnerable to attack this disease. The action of the project is a value crosses between olive varieties, new genotypes resistant to Xylella, increase their capacity to adapt to climate change, identify best practices and sustainable technology, increase biodiversity, and reduce water consumption, carbon footprint, involve multidisciplinary actors in the international cooperation that provides a new strategy for this prevention of Xylella fastidiosa and the option of UA environment politics and policy. And now the partners is Balon Agritur is the resident in Spain, is a farmer in Carmona, is near of Sevilla, is Greenfield, Agri-Food, University of Cordoba, Conselho Nacional de Research of Bioeconomy is Toscana, and Philip Berrio is a farmer in Italy. Cerqueirão is a farmer in Portugal, and Balon Agricultura is a farmer in Spain. Now, the problem of the soil is a good problem, and 30% of the soil are in the productivity is the result of the FAO. Now, the soil, because the soil is very structured, no, is when this life is soil, the life is in the soil, and the soil is very structured. It is very important because the soil analytic methodology is macro and micro, but the largest amount of nutrients are not detected by routine soil analysis, is underground, not superficial. It's very important to look that lies and forms of nitrogen and farming or cropping system. The crops is lucerne, beans, clovers, because these plants is noduling bacteria. See as rhizobium is rhizobium and using azospirillum, using composting. There are many different species of bacteria and fungi that can populate in the soil. It's very important. And now, what is important? Biodiversity of the species, thermoregulation, organic matter, because in these countries, in Portugal, Spain, and Italy, many things of the soil is organic matter very low. Root development, population, mesofauna, and mist regulation. And one thing I thought the thermoregulation whereas when we have cover crops is very different thermoregulation in the soil. Tilled soil, how vulnerable is expecting, I don't know, because the farmers now is very old and different mind of thinking, and tillage soil are vulnerable. Soil crusting, wind, water, tillage, erosion, temperature extreme, water extreme. Is very difficult to speak and say the farmers, but don't move the soil. The right type of soil is very important, the structure is very important and different. Pluviometry is very important because tillage creates 5 problems: accelerate the soil degradation, accelerate soil erosion. It's very difficult to stay a conservation of soil in the olives, in the almonds, in the vine. It's very difficult to say a farmer and is a natural runs in the soil. I'll say roots is very important of different species. Now is a now is a key. What the name is? This is now is nabo is turnip. Unskol is in a brassica. Is very important for the root because the penetration of the soil is very important because this very water storage simply channel provide a bit with carbon. It's CO2 respiration is very important of the soil, the different types of the different species. Now is cover crops cocktail because it's important for organic matter and is important for diversity because the soil and the roots and the flowers is for auxiliary insects and another things for combat the Xylella fastidiosa. Carbon convert of the soil protection every day, every year. And another thing is very important because the crop biomass is the limit with you sequester of carbon, and plant biomass is very important. Only is living, only is dormant, only is dead. Is important for us because the temperature of soil is 20 graus. The difference if you have a cover crops or no is very, very important for us. And now is important this carbon and organic matter because this relation in organic carbon matter is that calculation, the measure of emission in the carbon is very important for us and for Europe with the sequester of carbon. The important ills of soil biology because the soil is good, the tree is good, everything is good if you are healthy. Is decompose biomass, recirculate the CO2 atmosphere. Pathogen is very important, improve water quality, water storage, water flow, water infiltration, because now the climate change is very important for us, the water, and carbon is the backbone of food security. Photosynthesis, free energy source, soil biology, the exudates is very important. The residues of protect against soil erosion, increase nutrient, increase soil structure, increase infiltration. The carbon and food security is very important, okay? And now the sun. The sun is power of life through the carbon cycle. And we need in this project with olives and almonds, not very different, but you speak in the another almonds, pecan, nuts, avocado, and 5 years is not very sun in this in the soil. Is very important the cycle of the soil. The soil is Earth's living skin, and sun is only energy for life on Earth. It's very important. The agriculture produce manage plant to cope capture soil energy and transformation in biological carbon that feeds our life on Earth. Is very important, the hours of north the sun. The carbon cycle is the photosynthesis is very important, but is biology when we have 12, 30 years, I speak with my professors with photosynthesis. We speak in photosynthesis many things, but the real what is photosynthesis is very important for us. And carbon energy flow is very important. The nutrients, what you don't see in the soil, is very important. The roots, the exudates, microbes, soil fauna is very important. Yes, the organic matter function, carbon the cycle with organic matter with corp biomass. When I speak with the projector in UA, he speak with me what you do the measure of the sequester of the emissions of the carbon. I say that in Portugal we have values and measure of quality of the sequester, and is the organic matter and crop biomass is one relation very strong, and natural fertility is very important, the biomass and the protector of the soil. And now organic matter is 58% of carbon, and crop biomass is 45, 46, 40. Is very important in this soil, because if you have a good soil, a good nutrients, you have a good plant. But the rule in carbon is biodiversity, is the conservation of the agriculture. Many problems in the olives and the almonds is maintain the conservation of the soil. Many rules. It is very important conservation the soil, increase stability of the system, increase the carbon storage, reduce nutrients, lose the groundwater effects, and more pollution with increased CO2 and nitrogen application. Increase resistance to exotic species is very important. More efficient and nutrients capture and decrease susceptibility to disease is a good soil, a good conservation of soil, and the biodiversity is very important in this matter. What is species? How number of species is very different, because various species is auxiliary of insects. Erosion is the soil. What the soil? The pH, the soil. What the pluviometry of the zone is very important. Plant species, various species, not only gramineans, leguminosas, brassica is very important. The different species and different families, because the functional diversity and composition is very important for this work in the insects, in the environment is very important, because the climate change is technology and farm practice these challenges, effect of soil tillage, effect of sequestered carbon is very important. This because is one more species, more soil and more roots, and this slide of multi-species cover crops is carbon is bottom line in is very different. Immediate yield increase, combat insects, break disease, mitigate climate extremes is very important in these benefits of the carbon. And the major farmers, the major association, you don't think the soil, and the soil is the pool of these moments. And this slide is very opportunity, because you know for the plants, but the roots is very important. In the roots you work in trichoderma, mycorrhizas is very important to rhizobium, is very important, and other things we in this project we need that heart of the field is the soil. Is very important because the new generation of the farmers, I think that see this another measuring the soil and another microorganisms. Is very important is the team of this live resilience. The team in Portugal is the team in Italy, is the team in Spain. It's very important of us because important this webinar to explain everything, and the work is many important. It's only the heart that one can see regularly, but is essential, is invisible to the eye. It is important these moments. And thanks, I speak very, very good. And Ben, thank you for this invitation.
Thank you for joining. Thank you very much. It was very interesting. I look forward to hear a lot more during the panel discussion, which will come up next. For this purpose, I will put the panelists on the screen. One moment, we have good. So again, thank you very much. This was very interesting to hear from the various projects. I would like to start with a question maybe to Mr. Saldarelli or Mr. Kompant. How can we make the products, any product, enter in the xylem to reach Xylella? What is your experience on the best method? Is it foliar spray, drip irrigation? What do you think works best?
Well, thank you for the invitation. In the frame of BioVexo, we tested a different way of application, including also endotherapy. We were not in favor of this because of the potential application to the farmers of the techniques. So we consider endotherapy possible, for example, for monumental trees that are also in Apulia, so justified. Therefore, in every case we tested, we were able to to inject our products, the two products in olive. We also tested before in potted plants, but the results in the fields were not so good. Really, the spray application of the same product was better than the same product applied by endotherapy. So we gave up, and you should consider that if you can on a larger tree, on bigger tree of even 30, 40 years old, you should inject for every branch. So at least 3 branches, 3 main branches to inject. So this makes the problem. The problems of each product that has been tested and is being tested with Xylella is to let the product to arrive to the xylem where the bacterium is present. We have evidences that in some cases, one of our product, the one that we are able to detect, PSJN, is present in the total DNA extracted even a few months later on the plants that we sprayed. We use a surfactant to facilitate this. In parallel, we also, these were also are new. In parallel, we have also tested the possibility to bacterize, I would say, the roots of a plant with PSJN. And preliminary results told us that there is the possibility that an olive plants takes the bacterium from the roots, and the bacterium moves along the apoplast. We have this kind of evidence that we should better define. So this could be useful, for example, for nursery treated plants that can be bacterized before being transplanted in the field. PSJN, if I'm not wrong, Stefan could help me, has been also the problem with PSJN is that it induces plant immunity. So it can kill itself when it is sprayed. And that's a problem on using a bacterium, an endophyte that induce a plant immune response. This was also demonstrated by Stephen Lindov on Graven. Another problem of PSJN, I think these results are encouraging because the way of application, mainly for bigger plants, can be improved, can be perfectioned to let the bacterium to perform its activity better. And finally, if I'm not wrong, PSGN is able also to work as an epiphyte. So it induces a plant immune response, even if it does not completely enter in the xylem. So inducing a plant immune response that can help the plants to control the bacterium. These are, Stefan perhaps could help, could add something about this.
Yeah, thanks, Pasquale. PSGN normally is very well known to be an endophyte. I have studied this during my PhD and during several years. But when we apply it as foliar spray, it doesn't enter inside the plants. Why? Because it doesn't go to the xylem after foliar spray application. But what is more important, in our opinion, is that it reduces symptoms because of the plant immunity. And when we see also in the field, Pasquale was talking about endotherapy. We tested that. But what is the best solution for farmers? If the farmers is doing endotherapy, he need to buy the device to make the endotherapy, which costs. He cannot buy only one device for 10 hectares. He needs several one. And finally, the foliar spray or drip irrigation with some biopesticide is the more easiest way for application in the field for farmers, because the farmers has already the equipment for that. And of course, in BioVexo, I show you some result when there is a reduction of the spread of the symptom, but we don't have effect on the Xylella population. We cannot cure completely Xylella, but what we want to see is that something that can be relevant for the farmer to save his production The landscape also. It's money for living and to sell olives on the market. The farmer will need to live with Xylella, but if we can reduce symptoms in another way, which is costly and nice, costly, good, I mean cheap for the farmer, just to buy the product and to apply it in the field, compared to endotherapy when you need, I told you, the device. And imagine also with endotherapy, how many xylem vessels are present inside the olives. And when the Xylella, the insect vector, is picking the twigs, they are in the canopy of the olive trees. So even if you make endotherapy in the trunk, you're not sure you will reach the xylem vessels that are impacted by Xylella in the canopy of the plants. But we tested this solution because the European officer of BioVexo asked us to test endotherapy for that. In the case of some other plants, for example, grapevine plants, I know that endotherapy can be used against trunk disease when they have a nice biocontrol agent. But for olives, it's very different system. And what I told you, it should be cheap for the farmers and giving good result. Yeah.
Thank you very much. That was very interesting. Would anybody like to add with their experience? No? So maybe I would like to ask Mrs. Lander from BeXyl project, given the wide range of integrated strategy tested in BeXyl, from remote sensing on genomic approaches to biocontrol tools like bacteriophages, which do you think holds the most promise for scalable implementation across different European contexts, and why?
That's a difficult question to answer. Because we are focusing on biopesticides, so I will try to choose which one. That is difficult. According to the previous experience and some bioproduct that has been already registered, so maybe for vector control, it will be much easier to register some entomopathogenic fungi. For instance, the University of Cordoba has already experience in licensing some products, some strains to some companies, private companies, because some of the species are similar to others already registered. It could be much easier for developing a product that the farmer can apply. Also, because it is operated when you apply into the grass, to the nymphs, and also the plants, the endophyte, the entomopathogenic fungi can colonize endophytically the plant. So this could be also an extra benefit that it lasts longer, the product under the field. Concerning other bioproducts for the bacterium, some bacteriophages have been already developed for Xylella, but the market is not still very large. So maybe this could be something that has some future. And concerning the microbial synthetic community, this can be more difficult because since you have different species in the mixture, so maybe the regulatory process will be much longer. I know there are some mixture of biocontrol, not biocontrol, of microorganisms, but all of them are sold as plant promoters. Okay, so they are not as biofungicide or bio for biological control process. So maybe this can take longer. Yeah. Or maybe there is a need to of a second BioVexo project that you can implement the products we are developing now in BC.
Well, thank you very much. I would like to hand over to Ms. Migliotti. What can you tell us on how NextGenBioPest is planning to introduce biopesticides to farmers? And what can you foresee are the main challenges we see in this?
Thank you for the question. So actually, this is one of the main objectives of NextGenBioPest to actually communicate the products that we are going to develop to the final user, who is the farmer. During the project, there are some specific work packages that are about field trials and workshops and demonstrations. So we have already begun to do so. Actually, in June, we had a workshop in Montpellier where we introduced some first information to the farmers. And our next one will be a living lab in October in Greece, in the field in Aliartos I mentioned earlier. So through these living labs and workshops, we are going first to introduce the new tools in order to raise awareness as a first step. And then as the project progresses and we have actual results, we are going to help the farmers use them, inform them about the value of doing so, etc. However, what we have seen until now as a main challenge is, of course, the lack of knowledge, and this makes the farmer not so willing to adopt new technologies and tools. Also, besides this, we cannot say that there is not a high upfront cost for adopting such technologies. However, through our project and through our practical trials, we try to make the farmers more willing to adopt such technologies and actually to learn how to use them, because biopesticides differ from the conventional pesticides in how to use them. So what we did is that we have a whole work package dedicated to these practical trials.
Thank you very much. At this point, I would also like to remind the audience, if they have any questions, please just write in the chat function your questions, your thoughts, and later on we will be happy to answer your questions. Mr. Abreu, I would like to ask you, what are the main challenges in implementing biodiversity and sustainability in the olive sector?
Is a good question. But the project go to environment benefits of the mix of the cover crops, of the ecosystem service. But the problem of the farmers is one measure this ecosystem service, because carbon sequester, structure of the soil, fauna, biodiversity, how measure how many I pay. And now the UA in the sequestration of carbon is now in community 2020 weight, expect the pay the sequester of carbon. And is very important for us is one problem is the age of the farmers is very old. Is change your mind is very difficult. It's very difficult to change the new techniques that accept. Is the olive trees compete with the cover crops or not compete of the water of the system. Is very difficult to explain to the farmer the good problem is the soil mobilization, because I have in various farmers mobilization of the soil is a problem, because not conservation, not organic matter, not microorganisms, is very difficult to change these new techniques. Another change is in the market, you have a certificate of the olive trees with the biodiversity, sustainable, and sequester of carbon. Is when the certificate is on the market, is more easy for us work in the farmers with combat this Xylella and the trees of olive, of almonds. And now I think that the big problem is the new generation in this is a problem, because not open mind. And I think that the better the future is many information. And Miliotti is a new, because my farmers not email, not for very problem of the internet is a problem, is the difficulty of us.
Mm-hmm. Would anybody like to add to this? What's their experience with farmers taking on new technologies?
I think that for the extensive crops and the tree crops is quite difficult. So normally farmers that produce horticulture crops are more prone to be innovative and to acquire new developments, not only in biopesticides, but also new varieties and new crop systems. For my experience with olive growers and almond growers is that they are more conservative and more resilient to the old approaches they have been practicing for years. Yeah.
Thank you. I think we have time for a very fast second round of questions. So I would like to start with Mr. Saldarelli or Mr. Compant again. Considering that up to now there exists no product that can eradicate or eliminate Xylella from infected plants, particularly olive trees, how can we fix an efficacy threshold for a bio-based product? In other words, what is the level of acceptance of farmers for a product that does not eradicate the bacterium?
That's a difficult question that I propose initially. This question came from the fact that we are not able to eliminate the bacterium from infected plants. So in the reality, we are also able to exploit this, I would say, the side effects of the bacterium, which are the stimulation of plant immunity and also an activity of plant growth promotion. So this makes in such a way our products, our product development something that is not really a biopesticides. As Blanca said before, it's a biostimulant in that form. So this is a question that I pose to myself. How can be accepted a threshold, and how can be fixed the threshold for farmers to accept a biostimulant and introduce it in the current farmer olive culture cultivation. So I have not an answer for this. I don't know what I can do. Also because it is a process that takes really many years to be the evaluation of the efficacy or not of a product takes a lot of years, and also considering, for example, the benefit or not on the yield of the production, something that we have not considered in BioVexo, but it is of course a major point that that should be considered to introduce biopesticides in the current management in the infected area that we have in Apulia. There is another field of activity that we have not considered is that the possibility to use also this biostimulant in resistant cultivars that are now allowed to plant in the infected area and that now constitute the majority of the olive cultivation in the epidemic area. So I have not an answer for this. This will be this will be something that will be perhaps need also more from the other side, instead from the side of the biopesticides, again the vector, there could be our biopesticide seems to work on juvenile and also on adults, but is not persistent. So this perhaps however, these biopesticides could be better accepted in organic farming in this way of cultivation where for Xylella now I think there is only a product that is allowed which comes from orange. I don't remember the name of the product. So perhaps the biopesticides against the vector could be better accepted in the practical management.
Stefane?
Yeah, thank you. I would like also to ask question. What could be acceptable for the farmers? To lose the whole olive production, to sell the terroir where the olive were grown since many generations to a very low price, or to use some solutions that can reduce the spread of the symptom. For the moment, there is nothing at all on the market. Maybe in the future there will be, and I know there is some development for that, more tolerant cultivar. But if we can reduce the symptom spread in the next future as soon as possible, will be acceptable for the farmer. But of course, we don't have miraculous solutions. There will be not a reduction of 100% of the progress of the symptom. What could be acceptable for the farmer? We need also to ask the farmer and to learn from the farmer, and not only from the scientist, what could be the solution that can be acceptable for all. But you know, at the same time, in BioVexo, we are developing some biopesticides. There will be some products that will be on the market in the future, but maybe not all of them, because there will be, for registration, it takes a lot of years and it takes also a lot of money for that. So we are developing some solution, but we don't know if all the solution will be on the market for the farmers. And I can tell you, to find something on the market as a solution, it will take, even if you develop in a project like BioVexo, it can take 7-10 years before to be on the market. Then we need to have a very good solution. I show you some result when in San Vito de Normani, we have a good reduction of the symptom progress after 4 years of experiment. But in another one, there was a positive effect at the beginning and then not at the end because of the high pressure. I think what can be acceptable for the farmer is to have multiple solutions in an IPM strategy to reduce really more and more the spread of the symptom, the Xylella population, and to combine different solutions, combining reduction of the insect vector, Xylella, also with cover ground management, and put also different cultivar more resistant. We did not try to see if our biopesticide has an effect on more resistant cultivar, unfortunately, because we cannot do everything in 5 years and a half. We just provide some knowledge a bit, try to develop some solution, but I'm sure in the future there will be also some other project to reduce more the Xylella problem in Europe, which is very urgent. But I told you also for registration process, it will take time and money, and you need to have a big player for this big company to do this, and also to combine biopesticide with biostimulants and a lot of different solutions.
Thank you, Mr. Compound. I would
You
like
can
to
call me
ask.
Stefan. You can call me Stefan. You know me.
Thank you, Stefan. I would also like to ask the panel, but also the audience. Would you have another question? Actually, you would like to answer here? Do you have something to add to what we've heard so far? If not, then I would like to hand over to Mrs. Martin. 1 second.
I can unmute myself. I'm speaking. Thank you, Ben. Thank you. Thank you very much. Thank you, everyone, for this great webinar. I hope it's a really pleasure to host this webinar here today and hear from such a great mix of experts and projects. And a big thank you again to all the BioVexo partners and speakers from BeXyl, from NextGenBioPest and Life Resilience. We hope we sparked some few new ideas and give you some clear picture of the work being done across Europe and what we have planned even for future collaborations. And before we close the meeting, just a quick thing that I would like to share with you, if it's possible, is please, I'll do it now. I don't know if you can see my screen, but it's a quick reminder for save the date, please. Yeah, we're excited to inform you all about and invite you all for the BioVexo final event. It will take place in November 13. It is in Apulia in Italy. It's a full day event and full of project results and expert panels and dedicated sessions, also between sister projects, between projects, including some of the ones that are here today. So it's a great opportunity to meet in person and share some ideas. And we will share soon, as I said previously in the beginning, we will share soon some inputs in our social media, including the agenda and logistics and registration links and etc. So make sure to follow us on our social media. And one last thing before we go, I would like also to share with you the slides. I hope I can change. Yes. So here I'll be sharing the slides with the website addresses with all the projects that were featured today. So if you would like to dive deeper into their work and just have a screenshot of this slide and stay connected to learn a bit more about them. And once again, thank you all for taking part. A special thanks to Ben Leitner from RTDS Group and for making this session to happen. I'm not sure if you would like to say something before we close, but thank you for all of you, and see you in Apulia in November.
Thank you very much. I would also like to thank the panelists for their active contribution. Thank you very much. And I hope to see you at the final BioVexo event in November. Thank you very much.
I will try to come. No, I'm joking.
Okay.
Thank you.
Bye-bye.
Have a good day.
Thank you very much, everyone.
Bye-bye.
Bye-bye. Thank you.
Bye.
Bye.